Dear students, in this episode, I, Dr. C. Anita, will talk about the ultra trace minerals, that is manganese, molybdenum and selenium. Their principal functions are as integral constituents of enzymes or as activators of enzymes. A diet that is adequate in other nutrients and that does not contain a high proportion of refined foods is considered to satisfy the needs of these elements. Dietary deficiency is not likely in human beings. After reading this episode, you will be able to discuss the distribution of manganese, molybdenum and selenium, explain the absorption and utilization, identify the food sources, and understand the consequences of deficiency as well as toxicity. For your convenience, this episode has been divided into 3 parts: manganese, molybdenum and selenium. Let us start with manganese. Although manganese was recognized as an essential nutrient in 1931, its unique role as a cofactor in a variety of enzymatic reacts was later appreciated when manganese deficiency in humans was first reported in 1972, and its efficiency was well established. Manganese is found to be a vital component in the synthesis of protein, DNA and RNA. It is abundantly found in plant food sources than animal source of food. Let us understand the distribution in human body. An adult man contains 20 to 30 milligram of manganese, which is primarily in bone, liver, pancreas and brain. Manganese is found in most organs and tissues including hair, although its concentration is highest in the bones, that is 25 to 40% of the total body stores, and liver, pancreas and kidneys. Let us understand the biological role of manganese. Manganese is both an activator and constituent of several enzymes. Manganese is a component of pyruvate, carboxylase and mitochondrial superoxide dismutase. Enzymes in the antioxidant defense mechanisms require manganese. Manganese is essential for the normal skeletal and connective tissue development. It activates several enzymes involved in the formation of cartilage in bone and skin. It is also associated with carbohydrate and lipid metabolism. Manganese also plays a role in the formation of enzymes involved in carbon dioxide fixation. Let us identify some of the food sources. Manganese is widespread among foods, but the manganese content of foods varies greatly. Plant foods like wheat bran, rice bran, barley, oatmeal and rye are rich in manganese, that is, it contains about 1 to 10 milligram per 100 gram. While fruits and vegetables like pineapple, beans and lentils are moderate source, while foods of animal origin like egg, beef and chicken contain very low levels of manganese. Tea is the richest source of manganese. Let us understand the process of absorption and utilization of manganese. Manganese is poorly absorbed from the gastrointestinal tract, with only about 3 to 5% being absorbed in healthy individuals. Manganese is equally absorbed throughout the length of the small intestine by a 2-step mechanism involving initial uptake from the lumen and then transfer across the mucosal cells of the body. After absorption, manganese is transported to the liver, where a small portion is bound to a specific carrier protein or transferrin. The absorbed manganese may either remain free or become bounded to alpha-2 macroglobulin and transported to liver and then enters blood in free form. Serum manganese concentration range from 0.6 to 4.3 microgram per ml. The primary routine of excretion is in the bile and a little appears in the urine. Manganese is predominantly in mitochondria, and thus organs rich in mitochondria such as liver, kidney and pancreas have relatively high manganese concentration. The concentration of manganese in tissue is accurately controlled by the promoting the excretion of the excess metal from the body. Manganese absorption decreases as the intake increases and increases as intake decreases. Female may absorb greater amounts than males. Manganese absorption depends upon low capacity, high affinity active carrier protein such as divalent mineral transporter. Both regulation of intestinal absorption and control of excretion enable manganese homeostasis in the body. Low molecular weight ligands such as histidine and citrate enhance manganese absorption. Detrimental effects on manganese absorption include iron, calcium, fiber, phytate, oxalic acid and phosphorus. They may precipitate manganese in the gastrointestinal tract. Copper also decreases its absorption and retention. Deficiency. Manganese deficiency has been reported in many species of animals but not so far in humans. Signs of manganese deficiency include impaired growth, skeletal abnormalities, disturbed or depressed reproductive function, ataxia of the newborn, and defects in lipid and carbohydrate metabolism. Glucose tolerance in man is reported to be due to manganese deficiency besides chromium deficiency. Toxicity of manganese. In contrast to other trace metals, manganese has a low toxicity. Intake greater than 1 milligram per gram of food will produce toxic signs. The most common form of manganese toxicity is the result of chronic inhalation of large amounts of airborne manganese in mines, steel mills, and some chemical industries. Signs of toxicity are manifested in the form of severe psychiatric abnormalities, including hyperirritability, violent acts and hallucinations, and dementia, followed by neurological changes similar to those of Parkinson's disease and Wilson's disease. The major signs of manganese toxicity in animals are depressed growth, depressed appetite, impaired iron metabolism, and altered brain function. Manganese has been shown to interfere with iron absorption. On the other hand, iron deficiency leads to increased manganese absorption. Thus, iron deficiency can make an individual vulnerable to manganese toxicity, whereas manganese exposure might induce anemia by inhibiting iron absorption. Selenium. Selenium is one of the newer minerals discovered during the 20th century as being both required and toxicity. The existence of selenium as a metal was first reported by J.J. Berlesians in 1817. However, its recognition as essential nutrient did not occur until Schwartz and Forts showed that a form of liver necrosis in rats could be cured if either vitamin E or selenium were administered. Distribution of selenium. Selenium occurs in all the cells and tissues in the body in concentrations that vary with the tissue and the level of chemical form of selenium in the diet. A highest concentration of selenium is in kidney, liver, spleen, pancreas, and testes. Cardiac muscle has consistently higher selenium than skeletal muscle. On an average, the normal adult has about 15 milligrams of selenium. Let us understand the biological role of selenium. Selenium is a constituent of several proteins, thus it is an essential constituent of the enzymes. Selenium is an integral component of the enzyme glutathione peroxides. Selenium and vitamin E plays an important role in preventing cellular damage by lipid peroxidases. Selenium also functions in the oxidative phosphorylation of energy compounds. Selenium also influences the metabolism and toxicity of certain drugs and chemicals. The toxicity of certain compounds is increased in selenium deficiency. It is also seen that selenium somehow binds with toxic minerals such as arsenic, mercury and cadmium and renders them less harmful. Let us identify some of the food sources of selenium. Meat and seafoods are rich sources of selenium. Vegetables and fruits are poor sources. Selenium is present in foods mainly as selenomethionine and selenocysteine. The level of selenium in individual foods of plant origin is highly variable and depends mainly on the soil condition under which they are grown. Fruits and vegetables are generally low in selenium. Cereals and pulses reflect the selenium concentration of the soil in which they are grown. Organ meats like liver, kidney, etc. are good sources. Fish, especially tuna and shellfish, are rich in selenium. The mechanism of absorption and utilization of selenium. Selenium compounds are generally very efficiently absorbed by humans, with most of the ingested selenium absorbed readily from variety of foodstuff. Although the bioavailability of inorganic selenium is less than that of the organic forms, this is probably of little practical significance because all the usual dietary forms of selenium are absorbed efficiently. Selenium is absorbed from the duodenum, cecum and colon in animals. It is transported actively across the intestinal brush border. Selenocysteine may get absorbed by amino acid transport system specific to cysteine. Absorbed selenium is transported bound to plasma proteins for distribution to the tissues. The excretion of selenium is largely by way of the kidneys, although small amounts can be eliminated in the feces. It is excreted in the urine as trimethylselenite and in the breath as dimethylselenide, a volatile compound with garlic-like odor. The latter appears in the breath only when selenium intake is high and is a sign of toxicity of the metal. The deficiency diseases of selenium. Non-endemic deficiencies of selenium. Biochemical evidence of selenium depletion is not uncommon in subjects maintained on parenteral or enteral feeding for long periods. Low selenium contents of some infant formula have been reported to reduce serum selenium. Clinical manifestations of deficiency arising from such situations are uncommon and poorly defined. They include muscular weakness and myalgia with the development of congestive heart failure. Cachon disease. In man, the most striking selenium responsive syndrome is that of Cachon disease, an endemic and often fatal cardiomyopathy that affects mainly children aged between 2 to 10 years and women of childbearing age. Cachon disease was first described in the Chinese medical literature more than 100 years ago, but not until 40 years after its widespread occurrence in 1935 was it discovered that selenium deficiency was an important factor in its etiology. Typical manifestations are fatigue after even mild exercise, cardiac arrhythmia and palpitations, loss of appetite, cardiac insufficiency, cardiomegaly, and congestive heart failure. Pathological changes include a multifocal myocardial necrosis and fibrosis. The coronary arteries are especially unaffected. Ultrastructural studies show that membranous organs such as mitochondria or sarcolemma are affected earliest. Recent studies indicate that geochemical variables have an important influence on the distribution of Cation disease. Acid soils high in organic material and iron oxide content appears to be responsible for fixing selenium in forms that are poorly absorbed by staple crops. Cation-Beck disease is a selenium responsive bone and joint disease condition involving severe joint deformity which affects children between 5 to 13 years of age. The disease is characterized by joint necrosis, epiphyseal degeneration of the arm and leg joints, resulting in structural shortening of the fingers and long bones with consequent growth retardation and stunting. Although not identical to Cation disease, Cation-Beck disease also occurs in areas where the availability of soil selenium for crop growth is low. The selenium contents of hair and of whole blood are abnormally low. Cartilaginous degeneration is prominent and it has been suggested as another form of selenium deficiency. States of mild to moderate selenium deficiency have been proposed as contributing to a large number of conditions such as cardiovascular disorders, infertility, aging, eye disease, diabetic retinopathy, and cancer. Although a firm link between selenium and these diseases has not been established, there could be the potential involvement of a mild impairment in antioxidant activity with reduced luteolin peroxide levels. Selenium status and susceptibility to infection. As mentioned previously, expression of the cardiac lesions of Cation disease probably involve not only the development of selenium deficiency but also infection with Coxsackie virus. The early preclinical stages of development of human immunodeficiency virus infection are accompanied by a very marked decline in plasma selenium. However, for the nutrients affected, there are strong indications that only the extent of the decline in selenium status has predictive value with respect to both the rate of development of AIDS and its resulting mortality. The virulence of other RNA viruses such as hepatitis B and those associated with the development of hemolytic anemias are enhanced similarly by a decline in selenium status. Selenium and thyroid hormone. The importance of selenium for thyroid hormone metabolism is evident from changes in the T3-T4 ratio which develop after relatively mild selenium depletion in infants and elderly subjects. Decreases in T3-T4 ratio indicative of decreased thyroid hormone balance have been detected when serum selenium falls between 0.9 millimolar per liter. Toxicity of selenium is called selenosis, has been observed both in minors and in people who consume excess selenium supplements. The upper limit of a safe intake of selenium is 200 microgram per day. Excessive selenium produces cirrhosis of the liver, enlargement of spleen, gastrointestinal bleeding, and depression. Signs and symptoms of toxicity include nausea, vomiting, fatigue, diarrhea, hair and nail brittleness and loss, paresthesia, interference in sulfur metabolism, and inhibition of protein synthesis. Mottled tooth, enamel, and caries are endemic in affected areas. Now we will talk about molybdenum. The need for molybdenum was established in humans through the absorption that a genetic deficiency of specific enzymes that require molybdenum as a cofactor resulted in severe pathology. Distribution of molybdenum. In the body, 2 mg of molybdenum is present. Molybdenum content of human tissues is quite low, averaging 0.1 to 1.0 microgram per gram of wet weight. The liver, kidneys, and bones contain the most amount of molybdenum. Other tissues such as small intestine, lungs, spleen, brain, thyroid, and adrenal glands and muscles also contain the element. In tissues, it is present in 3 forms: molybdate, free molybdotrin, and molybdoprotinin, that is bound to enzymes. Let us understand the biological role of molybdenum. Molybdenum functions as an enzyme cofactor, and it is also be involved in stabilizing the steroid binding ability. Let us identify the food sources of molybdenum. Molybdenum is essentially for plant and animal life. Molybdenum is widespread in plant foods, and the content may vary greatly depending upon the concentration in soil. Molybdenum is found especially in legumes, meat, fish, and poultry. Provides up to 129 microgram per 100 gram. Whole grain cereals contain 117 microgram per 100 gram. Nuts and vegetables are moderate source, while fruits and dairy products are low in molybdenum. Describe the process of absorption and utilization of molybdenum. Molybdenum is found in foods primarily do not require digestion, as it is in the form of soluble complexes which is readily absorbed. Humans absorbed 88 to 93% of molybdenum as ammonium molybdenum in liquid form in the diet. The mechanism by which molybdenum is absorbed is thought to be passive with a carrier. It is absorbed as a molybdate and is concentrated especially in the liver, adrenal, and kidney. Transportation of molybdenum in blood occurs as molybdate, which binds to albumin. Deficiency of molybdenum. Molybdenum deficiency in human is not known. However, the deficiency was observed in human disorder caused by a lack of functioning molybdenum present in sulfitoxidase, a lethal inborn error in metabolism resulting from a sulfite oxidase deficiency is characterized by severe brain damage, mental retardation, dislocation of ocular lenses, and increased output of sulfite. Toxicity of molybdenum. In humans, both occupational and high dietary exposures to molybdenum has been linked by epidemiologic methods to elevated uric acid concentration in blood and increased incidence of gout. Molybdenum competes with copper for the same metabolic sites, and an excess of molybdenum will result in symptoms of copper deficiency. Students, let us sum up today's episode. Manganese is found to be a vital component in the synthesis of protein, DNA, and RNA. It is abundantly found in plant food sources than animal source of food. Manganese is poorly absorbed from the gastrointestinal tract, with only about 3 to 5% being absorbed in healthy individuals. Manganese deficiency has been produced in many species of animals, but not so far in humans. Selenium is one of the newer minerals discovered during the 20th century as being both required and toxicity. Selenium occurs in all the cells and tissues in the body, in concentrations that vary with the tissue and the level of chemical forms of selenium in the diet. Selenium compounds are generally very efficiently absorbed by humans, with most of the ingested selenium absorbed readily from variety of foods. In man, the most striking selenium responsive syndrome is that of Cation disease, an endemic and often fatal cardiomyopathy that affects mainly the children aged 2 to 10 years and women of childbearing age. Cation-Beck disease is also a selenium responsive bone and joint disease condition involving severe joint deformity which affects children of 5 to 13 years of age. Environmental conditions and agricultural practices have a profound influence on the selenium content of many foods. Geographic differences in the content and availability of selenium from soils to food crops and animal products have a marked effect on the selenium status of entire communities. Molybdenum is found in foods primarily do not require digestion as it is in the form of soluble complexes which is readily so absorbed. The need for molybdenum was established in human through the absorption that a genetic deficiency of specific enzymes that require molybdenum as a cofactor resulted in severe pathology. Molybdenum deficiency in human is not known in human being. However, toxicity is evident in occupational exposure to the animals. Thank you, students. Hope that you have understood about these ultra minerals in today's episode. Thank you once again.